Replacement is the substitution of living animals by in vitro techniques like cells or tissues, computer models, lower organisms like bacteria or yeast or invertebrates, human studies. Sometimes that's possible, for instance in nutritional studies. People might use humans. The field of molecular biology, genomics, proteomics, etc. And there might be a long other list. And the list is also growing, I think, over time. Reduction means a decrease of the number of animals. And we can achieve that by standardizing our animals in terms of the genotype, for instance. So when you use inbred animals of the same genotype, you decrease variation. Comparing when you use outbred animals, for instance, having a larger gene, a different gene pool. So we also can standardize the animals in terms of microbiological quality, meaning that you have in your animal room animals from only one source, from only one manufacturer, only one breeder, instead of having them from different sources, which also might increase variation a lot. And that will, well, then you will have to use more and more animals per group, so to get your statistical significant results. We can also control the environment. So we can control the nutrition we give. We give them pellets. We don't give them other foods usually. So we give them the same pellets. It will never be exactly the same because they are compounds from the field. So there will always be some variation, but at least more or less controlled. We can control the climate in the animal room. We keep it on constant temperature. The same reason to decrease variation. And we used to control the cage as well. From the 50s onwards, we had our animals in a shoebox-like cage with bedding, and that was it. It's simple, and for all animals the same. But we found out that then we create more or less abnormal animals, because these animals cannot perform any species-specific behavior. They cannot make a proper nest, they cannot hide, and they are prey animals, so in the wild they will immediately hide, and they still have that inbuilt, as an inbuilt instinct. So we deprive them from a lot of tools to perform species-specific behavior. And then you might end up with an animal which is showing abnormal behavior, like bar gnawing, or jumping all the time, that's typical, what we call stereotypic behavior. So, and then, of course, well, we cannot say our animals are completely normal. So we came back from that idea to standardize in that way. What we are now saying is that we should give the animals at least some, as we call it, enrichment or refinement of the environment, so that they can hide, that they can sit on top of shelter, or on top that they can climb, things they do also in the wild. And we can do that even in a sort of controlled way. It is not that we throw into the cage a lot of things, no, but we can give them a certain enrichment, as we call it, from which we know what it does to the animal. Whether it is beneficial to the animal, whether it increases variation or not, and in most cases it does not. So we think a little bit differently about the barren cage. But still in neuroscience you will find a lot of people saying, I don't want these things in the cage. I don't want it. I want a simple only bedding. And then you can talk and talk and talk. That will, yeah, it is very difficult sometimes to convince people that having abnormal animals, abnormal behavior animals, that will be no good for the experiment anyway. Okay, but we will talk about that later in the course too. We could control our experimental procedures to do always the injections in the same way, by preferably the same person, so that you don't get unnecessary variation in that part as well. And statistics. You will hear a lot about statistics because that's really important. You should do statistics prior to your experiment to estimate the number of animals per group, and not take a paper from 6 years ago saying, oh, they do more or less the same, so they used 6 animals per group or 10 animals per group. We do the same. No, you should calculate it for yourself, because it is, of course, it is bad when you use too many animals, which when you do your statistics afterwards, you might find out that you could have done it with much less animals. But on the other hand, it is evenly worse when you find out that you used too few animals, because then the animals you used, they are wasted anyway. So it should not be too many, it should not be too few. It should be the right number of animals to get your statistically significant results. Okay, refinement, meaning decreasing discomfort. So then we translate the biological needs of the animal into husbandry. I will show you in a minute. We might apply anesthesia and analgesia in painful experiments. Also, our skills are important. So before you go to your experimental animal, you might practice some surgery on dead animals, or, well, at least be sure that your skills are appropriate to the procedure you do. We can improve our procedures, so make the time a little bit shorter that the surgical procedure takes, that sort of things. And we should use a humane endpoint and provide adequate care to the animal. So, when we talk about translation of the needs of animals into husbandry, you can, of course, it's obvious that it makes a difference whether you house a rat, for instance, in a barren cage, only provided with some bedding, and that's it. Or that, for instance, when you have mice, that you give the mouse a piece of paper where they make a very nice nest out of it and they feel secure. So that makes already quite a difference. It makes also a difference whether you house your social living animals individually, like rabbits, for instance, here in individual cages, or that you house them in pairs. And now, according to our European rules, we have to give the animals, well, at least they have to be housed in pairs or in groups, but in pairs is possible. And then we have to give them a raised, so-called raised area, where they can sit on top and also hide underneath. Because also these animals are prey animals, so they like to hide, but they also like to sit on top. And when you watch rabbits and hares in the wild, you might see them sitting on their hind legs, looking around, because that is their way to control their environment. And they will still do that in the laboratory, although there will no predators present, but still they do. So they sit on top, and they like to be on a higher place. So for that reason, we give them that. Or we can also group house them. We can group house them like this, for instance, where you can see that they are socially housed, but also they have places where they can hide from each other. So you give them also separations and hiding places, as you can see. They have hay, at least, well, they have a lot of things to do, and also they are housed in a group. So that makes quite a difference how you house your animals, and that will have a reflection also on your experimental outcomes. How could we reduce discomfort? Well, first of all, we can question our own experiment. So we have to ask ourselves, do we think my project or my experiment is ethically admissible? We could choose another animal model, for instance. in some models, like in arthritis, for instance, when you induce arthritis in rats, they will develop those swollen joints, which is very painful. You might also use mice, where you might also use another induction method, but mice seem, at least, to suffer less from an arthritis model than the rat does. So when it is possible, you can switch to another model. You can also use another induction method to induce the arthritis. Well, in most cases, you have an adjuvant arthritis where you inject an adjuvant, like Freund adjuvant, to induce arthritis in all the joints. But there are also other possibilities to inject the animal with another substance, which is less painful, give less ulcers, and then the same disease is developing. We could, of course, use in vitro tests. Well, we could do imaging in long-term studies. So instead of killing the animals on certain time points, we can use imaging, and then we can do a longitudinal study like that. Computer simulation, we should use at least healthy animals, give them a stimulating environment, use anesthesia and analgesics, as I said before, develop our skills, give them adequate care, and also use humane endpoints. Well, what are humane endpoints? Well, humane endpoint is to define a set intervention point which allows the collection of quality scientific data, but limits the amount of suffering for the animal. Meaning that not the end of your experiment is the end of the animal, but that before that, when the animal gets ill or is not really feeling bad, you might say, well, this is the end, this is the humane endpoint, because I have collected my data, there's no need for further suffering. And you have to keep in mind that using sick animals to collect data might also not be good. So, and you can, as a researcher, you should be able to define a certain endpoint from the beginning already, before you start your experiment. And, well, some humane endpoints have already been established, like, for instance, when our animals show 20% weight loss of the normal weight, that could be, well, then you should say, okay, this is enough. Or when they lose weight, 50% weight within 2 days. But the problem is, of course, when you induce a tumor in the abdominal cavity, the tumor might grow and mask the loss of the body weight. So then we use the body condition score. And tomorrow I will explain what that is and show you how you do that. You might see a drop in body temperature over 4 degrees. That's also a reason to kill the animal. Or the tumor volume in a mouse. Well, there were some papers showing that it should not be over 1.2 times 1.2 centimeters, and in the rat 2.5 times 2.5. Also, when ulceration of the tumor occurs, well, that means also that you should kill the animal. When ascites is over 10% of the body weight, ascites is the fluid developing inside the abdominal cavity, so your animal gets like that, then also that's a reason to kill the animal. Or diarrhea, persisting diarrhea or incontinentia over 48 hours, or abnormal behaviors. Well, there can be a long list, of course. So, besides legislation, it is also important that international bodies provide guidelines and recommendations. For instance, we have, and Harry will also talk about that, we have global guidelines, like from the ICLAS, from the International Council on Laboratory Animal Science, or within Europe, the FLASA, the European Federation of Laboratory Animal Science Association, the UFO, the University Federation of Animal Welfare, the European Science Foundation, and in the US, and we have in a lot of countries, we have those bodies. Within Europe, we have the ESF, and that's the European Science Foundation, where a lot of science associations, like the Federation of Pharmacology, the Neuroscience Association, they are member of the European Science Foundation. And of course, well, they have the aim to advance the cooperation of scientists within Europe, or promote mobility of researchers, but also to advise national member organizations on science policy issues. And they wrote a position paper in 2001 recognizing the intrinsic value of an animal, meaning that an animal on its own has an intrinsic value, a value of its own. And that is contrary to the instrumental value, meaning the value which an animal has for ourselves, for our sake. So they recognize that. They endorsed the 3 R's, and they also supported the review of protocols by animal ethics committees and education and training. So that brings me to animal ethics committee only briefly, because also Harry Blom will tell you about that. What do? They perform an ethical evaluation, meaning that they outweigh the benefit or the relevance of a certain experiment against the suffering of the animal. That is the task. That's the main task of an animal ethics committee. Besides that, they also look at the implementation of the 3 R's, and they also look into the competence of the persons performing, designing and performing experiments. That brings me to the last part, to education and training. And I always like the quote of Peter Medawar, already an old quote, but still very valid to my opinion. The welfare of animals must depend on the understanding of animals, and one does not come by this understanding intuitively. It must be learned. That means that education and training in laboratory animal science is needed. And at least in Europe, and I hope also here, it's required for those persons taking care of the animals, the so-called category A, persons carrying out experiments and technicians, that's the so-called category B, and persons like you responsible for the design of experiments, so scientists, the so-called category C. And then we have also category D, meaning that those are the persons who have specialized in laboratory animal science at the postgraduate level. So, and that is also in our directive, in our European directive, it's saying that member states shall ensure that the staff is adequately educated and trained to do those procedures. So, we have done already many courses in many countries. This is a picture from a course we performed in Sao Paulo, in Brazil. So you see here two participants doing oral gavage in a rat, and that's exactly what you are also going to do in this on the practicals here. Okay, so why are laboratory animal science and the 3 R's important? For animal welfare reasons, scientific results reasons, legal obligation, and the judgment of ethical admissibility. Well, the last two we have discussed already, and I said at the beginning, the well-being of the animal and the scientific results are connected. And to show you that is by this simple sketch, so to say. When you perform an experimental procedure, there will be an intended effect that give you experimental results. But there will also be non-intended effects, leading to a disturbance of the homeostatic balance, and that on its turn might lead to compensatory mechanisms like changes in endocrinology or immunology, blood circulation, etc., etc. And yeah, that of course has non-specific effects on your experimental results. And those non-intended effects, yeah, what are they? Well, that might be pain, that might be the procedure you do, and housing. These things influence your experiment very much. So that means when you do it all well, that will have at least a good impact on your experimental outcome. So, therefore, I like to end with adding the fourth R to the 3 R's of Russell and Birch, the R of responsibility, meaning that our responsibility as a researcher is to ask ourselves, how important is it to use animals for this purpose? Can an alternative non-animal method be used? Can the number of animals be reduced? Can we reduce pain and discomfort? And what can be done to improve the living conditions of our animals? So, don't forget, happy animals make good science, and laboratory animal science can help with that. Okay, thank you very much. Any questions so far? Otherwise, there will be later, probably. Okay? Yeah.
Yeah, withdrawal of food means fasting for 16 hours.
Sorry?
Fasting for 8 to 16 hours. Is it considered a kind of stress for categorized into mild?
Yeah, well, your neighbor will talk a lot when he is teaching nutrition, but it depends what your aim also is. When you want to have an empty stomach, in the rat, for instance, the stomach is already empty after 6 hours. When you want to have an empty gut, it takes a little bit more time. But still, fasting will always have an influence on the behavior, for instance. Because they will look for food, they are much more active. There are also studies who have shown that. So, they will, at least, you see some behavioral changes. And it depends whether that might have an impact on your outcome or not. But you have to keep that in mind, certainly. Other questions?
Yeah, what we call post-invasive care. So, can we include it into degree of discomfort also? Like suppose we have performed some invasive operation on some animal, okay,
yeah.
And after that it would be under some post-operative care.
Yeah.
So it should be included into sort of discomfort for the animal, for say, for
certainly, certainly. Yeah, and you should include when you do invasive surgery, you should always give the animals, besides anesthetics, also analgesics. And of course, well, sometimes that is difficult because some of the analgesics might influence also your experiment. But then I think we always looked into ways to find the right analgesic which might not hamper your experiment. And also, what was said this morning was very, very true, that we must not forget that pain in itself will have a major impact on your experimental outcome. So, and that is not controlled. So, when you give your animal an analgesic, you know exactly the pharmaceutical, the pharmacon, you know the amount you inject, so it is a controlled analgesic. In pain, you don't know how many pain factors are circulating in the body. So that is much more variation, so to speak, and that is often forgotten, I think. So it's maybe better to accept the influence of the painkiller than accept pain. Yeah.
So now they talk of that fifth R, rehabilitation. Once our experiment is over, so who is supposed to take care of that animal? It's the responsibility of the person who's conducting or the organization.
No.
So rehabilitation is considered a part of the whole era what we are talking of, or it's a different issue altogether?
Well, you can argue that, but to my opinion, it's the responsibility of the scientist. It's your responsibility. You got the animal, and at least when your animal is still alive after your experiment, you could of course make agreements with the animal facility what to do with the animal, but you cannot say, okay, my experiment's over, okay, bye. No, I think you should discuss whether the animal can be used for something else, or that it has to be killed, or well, I don't know, but well, one of these two, I guess. But that I still, till the very end, it's the responsibility of the researcher.
So there is reduction of 15% of the body weight after 2 days. So should we try oral feeding or something like that, other than killing?
Depends what you want to do with the animal, because I can also see that after some experiments it might not be possible to use the animal for something else, because
So-
it might interfere with your other experiment. But you can but on the other hand, you can say, well, at least that's sometimes what we do in Holland as well. We could use the animal for training, the staff, the animal staff, for instance, or yourself in certain procedures. And that will be the last thing you do with the animal. So then that animal will be anesthetized, you practice some procedures, and then the animal is killed. That's better than simply killing the animal, that you use it for something else. But it depends entirely on the severity of the procedure you have done.
Can I comment to this?
Yeah.
Because there's also an argument that you can make that if you would only kill the animal without training on it, it would have less discomfort as compared when you would use it for training purposes. So if there is no need for training, then you probably better just kill it without causing more discomfort to the animal.
That depends what you...
So it depends if there is a need for training, then of course you best use the surplus animals for training purposes. But if there is no real need for training, then using it for training purposes would even inflict more discomfort on the animal than just kill it.
But when you do it under anesthesia,
Okay.
it should be done under anesthesia. That's what I mean.
It depends what kind of training it is. If you, it can also be trained. For instance, oral lavage, you can never do it under anesthesia, of course.
No, of course not. But it also depends what the severity of discomfort was in the first experiment. When that was already severe discomfort, you should not impose another severe discomfort procedure on the animal. So then it's better to anesthetize it and then do some training or something else. But well, there is a variety of possibilities at least. And well, the most important thing is that you think about it. Okay. Maybe you want to stretch your legs, but only for 5 minutes, and then we go on with the ethical part, because we have to install the movie in the meantime. So the next part will be we will talk about ethics before lunch. Okay?